Well, it's a real honor to be here. And every time I interact with Mike, I learn something. So what I learn is I'm older than he is. So what's wrong with me? What's my level of achievement? I can't compare, and I'm out of time now. So anyway. So Mike and I started working with Mike in 1980, and this was very close to when I started working in Alzheimer's disease. And at the University of Washington at that time, we looked around for somebody willing to do statistical genetics, and we didn't get anybody to bite. Then George Martin, the head of the Alzheimer's Center, called up Charlie Singh, and Charlie Singh introduced us to Mike Boehnke, and then the rest is history, as they say. So I'm going to talk about Alzheimer's disease. Everybody knows it's a progressive loss of cognitive function. The neuropathology which defines it are amyloid plaques, which are made up of the protein A beta, and neurofibrillary tangles on the right panel, which are made up of hyperphosphorylated tau. We now know a bit more in that there's a variable presence of Lewy bodies and alpha-synuclein, and TDP-43, which is a hallmark of frontal temporal dementia. So it's getting more complicated as we have better tools. Now, this is we have very good tools now to monitor different parts of Alzheimer's disease in living people. And the first thing that appears is amyloid deposition as seen by amyloid PET and CSF, analysis of CSF. This accumulation takes place at least 10 years prior before any clinical symptoms occur. So what we'd really like to be doing is studying this part of the disease where we can possibly intervene and possibly before the actual neurodegeneration begins to occur. So Mike and I did a candidate gene study with ApoC2. I say it's not my proudest paper, but it was early, and got me started working with Mike. And ApoC2 is part of a apolipoprotein gene cluster on chromosome 19. And Peggy Paracock Vance came along after us and cleaned it all up and identified ApoE as the actual gene. And I have to say, we were only 36 kb off out of a genome of 3 billion base pairs. That's not bad. It's not bad. E4 is the high-risk allele. E2 is protective. It's amazing that now, 30 years later, we're still trying to figure all of this out. There's a brain regulatory element, and all of the ApoC, or all of the ApoE that is in the brain is made in the brain. It's not transported in there. And recently, Jeff Vance identified an African specific haplotype that has a variant that attenuates ApoE4 effects. And I'll show you some of the ethnic diversity populations that we're working with. So that's beginning to pay off. We were also working on autosomal dominant early onset families, and this is a disguised pedigree. And this is a Volga German family. And they were 9 families that Tom Bird, another great collaborator that I've been able to work with, there are 9 families that came from the same 2 villages on the Volga River in Russia that were 5 kilometers apart. And these were the families that we used to find presenilin 2. And we also had early onset families that led us to presenilin 1. And Tom Bird was such a great collaborator in finding these families. And also, because we had these families, we could show that ApoE affects onset in the presence of presenilin 1 or presenilin 2 mutations. And presenilin is a protease that processes amyloid precursor protein and causes autosomal dominant Alzheimer's, but ApoE is also affects age of onset in these early onset families as well as in the late onset non-familial AD. So at this point, and I have to say this is a reunion of advisors. So all along, Mike was an advisor, as was Nancy Cox. And tomorrow we'll see Steve Rich. They were all part of that group that helped us along. So we came to the conclusion that collecting families was useful but expensive and difficult, and we kind of moved more to unrelated people. And in the literature, there were 10,000 AD cases. There were 10,000 cognitively normal controls, but they're all in different people's labs that were not collaborating. And so we had to convince NIA to support a large consortium to put all this together. And then we had to convince investigators to collaborate. I don't know how many times I've heard, well, we can't do it alone, except they act like they can do it alone. So anyway, so we convinced NIA to fund us to do the Alzheimer's Disease Genetics Consortium. And one of the key features of this, I'm talking type 2 diabetes to a type 2 diabetes crowd, but there were 3 papers in Science that put subjects together and were able to find additional or make a big impact on type 2 diabetes genes. And there were new methods for combining these data and showed the value of collaboration, and sample size matters. Not an astonishing concept, but it's still something we have to talk about. So these papers really helped convince NIA that they should be funding an Alzheimer's disease genetics consortium. So we started putting together samples from different groups, and the samples listed ADC 1, 2, 3. These are from the Alzheimer's disease centers. And these are centers across the country that all see patients with theoretically similar measures of cognitive ability and autopsy documentation. And this slide's a little out of date. It's probably more like 10,000 cases. And it's an extremely valuable sample because some of these people have been seen as many as 20 different times sequentially, and a tremendous prospective cohort that is of great value. Now, part of what was a little bit astonishing is these different groups were not collecting DNA. And this sample size is good, DNA is good. This isn't rocket science, but it took, even with Mike's status in the field, it took, I think, 3 years to convince NIA to say, centers have to collect DNA, okay? And then Barack Obama said you wanted how many millions of old dollars to go to Alzheimer's disease. Francis Collins said we needed to do sequencing, and then I won't go into the gory details that Mike helped push NHGRI into the process of doing this, contributing sequencing data. We also, at the same time, formed an international collaboration with the Charge Group and GRAD, which is UK, and EADI, which is French, and we're still collaborating with this group. It's difficult to get them to exchange individual level data, and I won't take a stand. Is that because there are legal barriers, or they just don't want to do it? So I don't want to say anything more. So since then, we've collected samples from all over the world, and we share all the data, so qualified investigators can get all of the data. And Lisan Wang runs the NIAGADS, which is the data sharing entity, and which also does a lot of other bioinformatics in terms of these samples, but that's critical for this whole process. And so at this point, he releases a whole genome sequencing data essentially once a year, and we're now up on R6, and that going to have 81,000 subjects and 64 cohorts that contribute to this sample size. Now, it's a highly multi-ethnic, so in getting samples from just the ADCs that were mainly non-Hispanic whites, but with a sampling of others, but now with a little bit more targeted recruitment, we've gotten the European slice much bigger, or the Hispanic and African American total bigger, and then on the right are the what's to come, and these are samples that are potentially available for sequencing. Now, it's been a little, I guess I was fairly naive about what Hispanic means, because a lot of studies link Hispanics as one population, and they're clearly not. So we have Hispanic Americans highly admixed with Mexico, people from Mexico, and these other Central South American countries. And so we have a very diverse population to work with. And they are very different in who they're admixed with. And some of the Caribbean Hispanics are admixed with African genotypes. There's the red slice on the left is the Peruvian. India are the La Ciudad samples from the Indian continent. And you can see that's quite a bit of difference in terms of this principal component analysis. That shouldn't be too surprising. Yes, none of the stuff I'm talking about is particularly surprising. Sample size matters, get DNA, pay attention to the ancestry. Okay, so what have we learned? So we've got anywhere from 90 to 120 loci where we don't know what the gene is. Maybe we know approximately 20 of those we can assign with a very high degree of confidence to a specific gene, so there's a lot of work to be done there. And I think there's about, well, people would argue with this, but I would say that there's at least 20 genes where you absolutely know that that is the gene that is responsible for the signal. The rare coding variants that are really useful in terms of figuring out what a particular gene does. So in a number of them, we found the TREM2 and a few others have been very useful in trying to figure out the pathogenesis of these genes. Now, I want to talk a little bit more about APP, and APP is both an early onset and late onset gene, and finally, brain inflammatory response contributes to AD onset and the progression of AD. So this is our latest collaborative study, and you see in the paper on the right, the authorship is just all these consortiums that are working together. And so when we get this quite large sample, we start to see APP as a late onset risk gene. Right next to it is another one, Adam TS1, which is a quite good candidate gene. It's a protease, but conditional analysis says these are separate genes, and they're separated by a TAD boundary, so we think these are just coincidentally two genes that are close to each other. So APP, the reason I show this is why is it important to know APP is also a late onset gene? And a lot of the drug development involves looking at A beta, the product of APP, and so if we develop a drug for based on that, will it work on late onset? And antibodies to A beta, which are the two approved therapies, work in late onset Alzheimer's. So it's very clear that what we can figure out studying APP is going to help us with late onset Alzheimer's. So this is another study we did, and this emphasizes the role of the brain inflammatory response in Alzheimer's disease. Now, for many decades, or several decades now, neuropathologists have said there are activated microglial cells in the brain, and the question is, are they just there to clean up damage, which is what microglial cells do, or what macrophages do, and when they're really activated, they kill cells, or are they part of the disease process? And I think when we did this study, we realized that we got four signals: TREM2, PLCG2, and ABI3, and these are all rare variants that are coding variants, and these are all microglial genes. And so this is a protein network that Peter Holman's put together, and all of the genes that I just showed you are part of this network, and all of the proteins in this network are overexpressed or highly expressed in microglial cells. And this just shows some of the expression data. So the top PLCG2 and ABI3 came out of this study, and as we go into the other genes that are known, SORL1 and the MSA6A cluster are highly expressed. So as the field progresses, we started to realize a lot of the genes that are coming up are microglial genes, are part of the inflammatory response. And this is from Brian Kunkel looked at in an ADGC study. He looked at what's the overexpression pattern, and this is using single nuclei sequence data. Microglial cells are highly overexpressed compared to any other type of cell, and the same pattern as shown here is also in all of the other brain regions that we have data for. So this is panel A and B are the single nuclei sequence data, and you can see that red blot on the left panel is microglial cells, and that's enriched in microglial cells that are clustered around plaques, and NCK2 in the second panel shows the same pattern. On the right, I want everybody to come up to look real closely at that one. That's all the genes in the large consortium study, and then the microglial column of the red blocks are the ones that show overexpression. And if you just take a distant look from that, you can see all the genes that we're coming up with out of the GWAS are enriched and highly expressed in microglial cells. And these data kind of evolved as people were doing GWAS, because, oh my gosh, a lot of these genes are microglial genes. And so I think what that tells us is, rather than microglial cells just swarming to clean up damage, they're actually part of the disease process. They're part of the onset of the disease. Now, pathway analysis, I always cringe a little bit in showing pathway analysis, and you always show pathway analysis when the pathways agree with what you think is going on. So this is from Brian Kunkel's work, and you see A beta and tau show up, as well as the activation of immune response. The same thing from the large consortium. Even more dramatic is, again, the second top pathway is the immune system. And there are 15 genes in that study that are part of that, but there's also 2,370 genes that are not. So please don't write this down. So here is a incomplete list of some of the immune genes that are showing up. The pathways are what we like to see: tau-related, amyloid-related, lipids, immunity, and endosome, lysosome pathways. Now, this is a very incomplete list of the drugs that are under development, and it's hard to, these are the ones that NIH is putting money into. And so it's not a comprehensive list of everything that's going on in pharma. That's very hard to get. And then there's overlaps between Alzheimer's and other disorders. So frontal temporal dementia, TDP-43, Lewy body, and in Alzheimer's disease, a 30 to 40% of people have Lewy body reactivity in the brain. Same for TDP-43. So that overlaps with Parkinson's and ALS and educational ascertainment. This is the group, right? Fortunately, it's protective. And then the last three, supranuclear palsy, cortical basal degeneration, Pick's disease, these are all tau-only disorders. So I've had this question put to me by Richard Otis, who is the head of NIH, and say, well, don't we have enough genes? And I was thinking on my feet when I came up with the answer. So I said, we have enough genes when we have a therapeutic to prevent or cure Alzheimer's disease that's inexpensive enough for third world, low research countries. And he says, oh, okay. So I like that answer so much. So Mike has been extremely generous with his time coming in and advising us. It's not just the advice, it's the advocacy to NIH and other people that's been important. So when I called Mike up and say, Mike, we've got another meeting, we need to come as an advisor. And he said, oh, come on, do I have to come? And then I say, well, no, you don't have to come. This is not a life sentence. But as soon as we have a therapeutic to prevent or cure Alzheimer's that is inexpensive to use in research-poor populations. Now, I want to come away with something from this meeting. So I have the Boehnke rules that I have accumulated, okay? What do you mean don't use the passive tense? I use that all the time. But sample size matters. Collaboration is great. Often publish early. And then the last two things you should not delegate, fatherhood and proofreading. Now, everybody here who's written a paper, huh? Everybody that's here has probably written a paper with Mike and had it come back with all kinds of, and so it's been it really helps. And then finally, he taught me this phrase, and he made me promise I would not use it frequently, that something is approaching statistical significance. And I have not used it yet. I've got it in my back pocket in case I need it, but this is one of the things Mike has taught me. So anybody that has more rules that would like to add, that would be great. And finally, I have advice to young people. Collaborate with people that are really, really smart. And the other thing is collaborate with your friends. Mike is a great friend. Thank you.